10-minute break, but like everything, we're all used to it. Conferences go over. We're just gonna, I guess we're just gonna go straight into our next session. And I'm gonna introduce, our second panel, of speakers. With that, our first speaker in the second session, which I know there was not a research pause for it, is Dr. ResearchGhjar. Hi, Cyrus, how are you? I think you're hopefully let him get on. I see Carrie.
Hi.
Oh, hey,
how are
you? Great to see you.
Good to see you. Nice to see you too.
Good.
Thanks
good.
for having me.
Of course. I think you have the access to share your screen while you put your slides up. I'm going to introduce you. So ResearchGhjar, is an associate professor of human biology and public health sciences division of the Fred Hutch in Seattle, Washington. He's the lab director for the laboratory to study metastatic microenvironments called LSM2. Very cool. Cyrus is, he's interested in studying how distant tissue microenvironments that we've been talking about, and then dormant cells, which are called disseminated cells, basically escape and elude, therapy, and they set up that environment that is necessary. He is one, again, of the leading researchers, and I'm always very fascinated and look forward to his talks. So Cyrus, with that, the Zoom floor is all yours.
Well, thank you, Josh, and thank you for the opportunity to participate. It's an honor and a privilege, and I look forward to the discussion and seeing everybody else's talks as well and the interaction afterwards. So what I want to talk to everybody about today is targeting dormant disseminated tumor cells for the purposes of preventing breast cancer metastasis. I want to start with an acknowledgement of my lab. This is as 2020 as a picture gets. We have a socially distanced picnic, and you can tell by the red sky there were fires in the Seattle area, a lot of smoke, so we were affected by all of that. But we have a wonderful lab, and we're back to full speed and working as hard as we can to make progress, because as important as it is to stay healthy and safe, it's just as important to advance our cancer research as well. So the metastatic cascade, I'm sure you've heard a lot about it in the first session, is typically depicted as this linear process where the primary tumor grows, it becomes invasive, and then eventually it enters the vasculature and then exits at secondary sites like the liver, the bone marrow, brain, lung. And what we're often taught in the textbooks is that if tumor cells can't survive at this secondary site, then they're able to adapt and proliferate to form metastases. This linearity of the metastatic cascade and the depiction of it in this fashion has really led to approaches where we try to target invasion, because it was thought if we can, block this step, it's the most genetically aberrant or mutated cells by the time a tumor is detected. If we can block them from invading, then we'll be able to prevent metastasis. And this has led to maybe a disproportionate amount of focus on trying to block processes that lead to invasion, MMP inhibitors, for instance. And by and large, these haven't really worked to prevent metastases. So we need more of a focus on what's going on at the secondary site. We need better therapies against metastases, but we also, I would argue, have a window of opportunity to do something about metastases before they arise. Because in breast cancer, we have these characteristic relapse peaks 18 months after treatment, 5 years after treatment, and then continued relapse after 5 years of treatment. And an increasingly large proportion of patients are relapsing later and later. So what do I mean by an increasingly large proportion? This is a study published in the New England Journal of Medicine at the end of 2017. And this is a huge retrospective analysis of about 70,000 women who were treated with 5 years of endocrine therapy. So these are estrogen receptor positive breast cancers, and these women were treated with 5 years of endocrine therapy. And what you can see when you look at recurrence as a function of lymph node status, no involved lymph nodes in this kind of orange, yellow color. I don't know what you would call that, mustard, perhaps. Tangerine, I don't know. You have some nodes involved in blue, and then extensive nodal involvement in red. And what you'll note is that you have recurrence 5 years, but if you look at recurrence going through year 20, the majority of recurrences are taking place after year 5. And what this is suggesting is that therapy is not curing these patients per se, but is putting off relapse into the future. And so I'd argue that we have this window of opportunity between the time the tumor is detected and the time that these metastases show up, maybe years or even decades later, to do something to target the seeds of metastases. And I'd also point out that currently we don't have a single therapy that's really aimed at selectively targeting the seeds of metastases. And this is, to me, a huge challenge and a huge opportunity to be able to really impact patients and prevent them from ultimately presenting with metastases. So what are we trying to target? From the time a breast tumor cell shows up in an organ like the lung to the time it becomes clinically detectable, what is it doing? Well, it could be doing a couple things. One is that it can exist as a dormant mass, so proliferation and death being balanced at roughly the same rate. The other thing that can happen is that it can exist as a cell, a single cell or a very small cluster of quiescent cells that have exited in the cell cycle and remain quiescent. And you can imagine, this isn't semantics. If you want to target a mass of maybe tens of millions of cells that are growing and dying at the same rate versus targeting a single cell that's quiescent, the biology that you're trying to target is going to be very different. So I think it's a very relevant question to ask what is actually present in an organ at any given time. And what we know the most about is bone marrow, because it's accessible. We can take a needle and put it into somebody's hip and actually suck out their bone marrow and stain for cytokeratin to identify cells of epithelial origin, cells that have to have come from the breast epithelia in a breast cancer patient and reside in the bone marrow. And by and large, this is what we find: single or small clusters of these red cytokeratin positive cells that are persisting in the marrow. And we know if they're there that they portend poorer metastasis-free survival, meaning these women are more likely to get metastases. This hasn't been studied in men with breast cancer, but I would venture that maybe we would find out the same thing. So is this what we want to target? Do we want to target these dormant cells? Are they really the cells that give rise to metastases later on? This is an assumption, and I'm very upfront about saying this is an assumption. And when I started my lab, this is absolutely an assumption that we wanted to test. And the way we went about that was with intravital imaging. So what this allows us to do is actually remove a piece of the skull and actually put a window on a mouse's brain. The mouse is perfectly fine afterwards. As you can see, if you did this to me, I don't think I would be perfectly fine with it. But mice just soldier on. They stay alive for weeks and weeks. They would stay alive indefinitely if it wasn't, for the fact that we're about to inject them with tumor cells. And what we can do, though, is inject them with fluorescent tumor cells. And now we can look inside of the brain, and you can see the kind of pictures we're able to take now inside of the brain to visualize cells arriving into the brain, these green cells in the red blood vessels, getting out of the vessels and actually setting up shop around the vessels. So what we wanted to do was take a line that could not form brain metastases and a line that could form brain metastases. And we wanted to make a comparison of where exactly the metastatic process fails in the cells that cannot form metastases versus those that do. And so I'm going to show you data for one line, and we've done this with two different lines, and we get the extremely similar results. So you can see that in one line that is not able to form brain metastases, these cells persist in the brain, and you can see single cells that are just aligned on the blood vessels and remain in the brain for over 6 weeks in this case. And then we have lines that will form. These are the same line. In this case, is MDA and B231, and then a brain metastatic MDA and B231 that we actually got from Patricia Stieg, who I see kind of underneath me to my left. Thank you, Patricia. Thank you, Pat, for this line. And so they form brain metastases. And now we've done this, as I said, with two different lines, T42 and MDA231. And then what do we see? What we see is that the latent lines in green and the brain tropic lines in blue, they don't differ in their ability to enter the brain. They enter the brain at roughly the same rate. They don't differ in their ability to survive in the brain, which is often called a very hostile site. They basically die at the same rate upon entry into the brain. Where they differ is in their ability to escape this phase of dormancy, progress towards micrometastases, and sustain those micrometastases. And what that argues is that this is a rate-limiting step, or maybe perhaps the rate-limiting step of metastatic progression, and the one that we want to target. And if we could target it, perhaps then we could prevent metastases. We could catch cells in the state and kill them. So what are the hallmarks of these cells? What can we understand about them, and what can we target? Well, one, we know that they survive for very long periods of time. And two, and despite being from the breast and existing at this distant site. And two, we know that they're growth arrested. And if we care about these cells, they must be reversibly growth arrested. They have to be able to wake up again. So what can we learn about that? Well, I note that one thing I want to point out is that each and every cell that gets into the brain, this isn't even a 999 out of 1,000 thing. We've actually tracked over 1,000 cells this way. Every single cell we've tracked actually sets up shop, not near the vasculature, but on the vasculature. And that's where they persist. And ultimately, no matter what they do, they have to be in contact with the vasculature. We don't note cells even ever leaving this niche. And this really is something that holds up regardless of where we look, whether it's the brain, liver, lymph node, bone marrow, or lung. And so what this has allowed us to do is actually try to understand these niches. We model them in culture. We create human vasculature in a dish, and then we study the interactions between the vasculature and the tumor cells. And we're able to recapitulate this phenomenon of dormancy or breast tumor quiescence in the dish. So a couple things that we've been doing is, number one, trying to understand what enables these cells to survive. We've run high throughput screens, meaning we are able to screen over 5,000 drugs in these microenvironments and look for drugs that selectively target dormant cells. And we've made a lot of progress in that area. It's actually very serendipitously making sense and harmonizing with another totally unrelated project we have in the lab. And we think that we've identified unique hallmarks of dormant cells that would allow us to kill them, perhaps without chemotherapy. We've also used these niches to try to understand what's actually putting these cells to sleep. If we could understand that, maybe we can actually just enforce these cues and keep these cells asleep indefinitely. The very interesting thing is that the mechanisms that seem to induce dormancy in lung and bone marrow differ from those in the lymph node, something that you would expect given that lymph node metastases are so common. What regulates those cells and keeps them dormant probably should differ from other organs. And they're totally different from what we've observed in the brain. In the brain, the burden actually seems to shift to perivascular cells called astrocytes, and they're inducing dormancy through a protein and through receptors that don't seem to matter at other sites. But a third hallmark of these cells is that they're chemoresistant. And so in the time that remains, what I want to do is tell you what we've tried to do to understand the fact that these cells that are asleep are resisting chemotherapy. The assumption has and really continues to be that cells that are not dividing are not sensitive to chemotherapy. So cells that are dormant don't respond to chemotherapy because they're dormant, not because of the environment that they occupy. We wanted to challenge this assumption. And the way that we challenged it was trying to understand if we treated mice with dose-dense chemotherapy the same way that a patient with invasive breast cancer would be treated with doxorubicin and cyclophosphamide, where would the cells that survive reside? So we looked in the bone marrow, we found disseminated tumor cells, and we mapped their distance to the vasculature and to other cell types. And the bottom line is what we found, that with chemotherapy, this actually selected for cells that were even closer to the vessel than they were to begin with. And you can see here, all these red dots collapse to the x-axis, and chemotherapy is really forcing these cells or selecting for the cells that are already sitting on the vasculature. So we wanted to study what it is on the surface of the vasculature that could be protecting these cells from chemotherapy. And we did a number of things, and we narrowed it down to some proteins that signal through molecules called integrins, specifically an integrin called integrin beta 1 and another integrin called integrin alpha V beta 3. And so what we did then was think about a way that we could actually trial this in a preclinical setting. We took a model of luminal B estrogen receptor positive breast cancer and delivered these cells into overectomized mice. So what we're trying to do is model estrogen suppression and progression under the context of estrogen suppression. And then we treated mice with this the adriamycin cyclophosphamide regimen, and then and either preceded that chemotherapy with just a generic antibody or antibodies targeting integrin beta 1 or integrin beta 1 and integrin alpha V beta 3. And what we did first was assess the burden of disseminated tumor cells in the marrow. And the second thing we did was actually see if this would help metastasis-free survival. So this is the bone of a mouse treated with chemotherapy preceded by generic antibody. This is the femur. You can see all of these green cells lit up in the femur and how much burden there is throughout the femur. This is the femur of a mouse treated with integrin inhibition in combination with chemotherapy. And now you can see that you see some green cells kind of scattered throughout, but they're really few and far between. So we've decreased the burden of disseminated cells in the marrow of these mice by 94%. And then the question is this actually going to lead to better metastasis-free survival? And the answer is that it does. And you can compare that the mice treated just with chemotherapy alone, over 70% of them recur with bone metastases and some with adrenal metastases. And that's in contrast to those where we preceded chemotherapy with this integrin beta-1 inhibitor, where only about 22% of the mice get succumb to bone metastases, or actually experience bone metastases down the road. And so the question is, okay, well, now we're kind of augmenting chemotherapy with an inhibitor to a very broadly expressed molecule. Does that have an adverse effect? And it doesn't in terms of body weight, in terms of vascular density, and in terms of bone marrow cellularity, where we see that, yes, chemotherapy has an effect on some of these metrics, but adding integrin beta-1 inhibition in the context of chemotherapy does not change that or enhance any of the chemotherapy-associated toxicities. So I will end there and say that we can conclude that the perivascular niche is protecting tumor cells from chemotherapy, and it is not because of their proliferation state, but because of the environment created in the vascular niche, and specifically through molecules deposited around the vasculature that are signaling through integrins on disseminated tumor cells, protecting them from chemotherapy. And when you interrupt these cues, you can now sensitize cells to chemotherapy. And the key is that you do this without waking them up. I didn't show you guys that, but we are sensitizing them to chemotherapy. And by inhibiting integrins, we are not actually waking these cells up or causing them to cycle. And this seems to occur, at least in mice, without undesired side effects. So the question is, we work with patient advocates quite a bit, and they're very adamant about the fact, I was really impressed with this, and I was very excited to show them the data. And I'm like, well, this is great, but we don't want chemotherapy. And this is why it's wonderful to continue talking to patients, because, I might have been satisfied with this result. Otherwise, now I'm actually not satisfied with this result. How do we remove chemotherapy from the equation? As I've said, we've screened for drugs that specifically kill dormant cells. In the absence of chemotherapy, I think we have some very, very promising leads that are potentially targeting new or at least previously unknown biology of these cells. And we're also now trying to discover immunotherapies and how to sensitize these cells to immunotherapies. And we are specifically focusing on CAR T cells for a variety of reasons that maybe I'll get into next time. So thank you for your attention. Thank you for allowing me the opportunity to speak here. And I'll look forward to the panel at the end of this.
Thanks, Cyrus. That was great as always, and very relatable.